You have no idea what I mean by that. I just realized.
That's from back in the day.
Yeah, I love Candy Cane. That's great. It's probably on YouTube.
Yeah.
And just as a reminder, you guys are going to be on the hook today. Lots of questions. Lots of people are going to have to engage. We want to hear what you guys have to say. It's important that you guys do engage with us. Please use your microphone if you can. Chatting is subpar. It's great to hear each other. It makes things better because we're already separated. And the more that we engage each other, the better off we are going to be academically and mentally. Also, if you do not have something to write with, you need something to write with today. You need to actually be writing information down. You're going to need to be taking notes. Otherwise, you're not going to be able to do the exit ticket very well at all. So you need to get something to write with. So if you would not mind, not if you don't mind, it's not a request, it's a demand. Go get something to write with if you do not have something to write with. You need something to write with and on to take notes. I'm not asking. It's like my daughter. I say, hey, can you do the dishes? It's not a suggestion. Do the dishes, man.
Okay, so we have one person with some tech issues.
Okie dokie. Okay. Now we're kind of in class phase. So you have two questions. So which equation is cellular respiration and photosynthesis? So type 1 is photosynthesis is on top and cellular respiration is bottom. Type 2 if cellular respiration is top and photosynthesis is the bottom. Oh my God, they've already started doing it. I didn't see it. Good job. Good job, DeLeon. Good job, Hudson. Good job, Jasmine. Woo! Yes, Sabina, you guys are our favorite class. Yes, yes, yes. Thank you, Gabe. Good job. Thank you, Bennett. Good job. So we've got 8 so far. I've got 8. I don't know how many you got.
I don't have any yet.
Oh crap! Send them to myself and Dr. Smith, both of us.
Okay. So I see
Don't be
Zach
insolent.
is coming by.
Awesome. That's going to be 9 because I don't have his.
And Hudson and Vo.
That's 12. There should be 19 of them. So we're waiting on 7 people.
Jasmine.
Awesome. We're waiting on the others. Don't make us call you out. Today is the day to get called out. Today is the day to get called out. You cannot casually be in class. You're not here just to talk. Just to watch. You're here to engage. So I have not heard anything. You tell me if you've heard anything from Soraya, from Jordan, from Hi. I don't have anything from them yet.
And I don't either.
All right, we're waiting on you guys.
And the ones I have, they are all correct. So
yeah, me too. They're all correct so far.
Hudson and Jasmine.
Excellent, excellent, excellent. Let's see, 1103. So if you're not entering the do now and you're not engaging, we cannot assume that you're here. It's going to be important that you, when we ask you a question, that you do engage. Otherwise, we assume that you're not here. For my juniors, I've already given them warning. This will be the only day I can give this to you. You don't chat back to us. You don't engage your microphone. I'm going to assume that you're not here, and you'll be bounced out of class. And you can watch the video, but you do need to engage the class. It's not just, you can't just sit and watch live. You can come back and watch the videos. You need to be here as a learning community. We got to do the best job we can. And you guys are right. And it looks like I've made a typo down here on my, I did put subscript for carbon here. That should be C lower, this little 6 should be a subscript. Oh well, I can't be perfect here. So 2 was correct. Cellular respiration is on top. We breathe in oxygen. This is a substrate going into respiration. We eat glucose. Glucose is released by plants. It's a product of plants. Oxygen is released by plants. It goes into the atmosphere. and then we recycle it. So these two things are going to be exchanged. There are substrates and respiration, and there are products in photosynthesis, and then we release CO2 in our breath that goes into photosynthesis. We release water when we urinate, and then that goes into photosynthesis. Light is going to take these two molecules and put them together to make glucose and oxygen, which we recycle. The energy is light energy getting converted to chemical energy. And then here we've got that same chemical energy being converted to another form of chemical energy. And some of it is releases heat and kinetic energy as well. But you guys did a really great job with that. So it looked like 12 of the 19 of you guys answered it. Good job to those of you guys. The 7 that didn't, you'll be amongst the first that get asked questions today in class, including now. So the cell cycle, when you look at this, we've got an image of the cell cycle here. What does this kind of remind you of? When you're looking at this image, what does it kind of look like the cell's doing? What does it seem like it's doing? And this is open for everybody. Please throw it out there openly. Please throw it out there to everyone when you answer it. Just I want to get your ideas. What does it look like the cell is doing? Mario, good job. You said, look, it's reproducing. Vu, looks like it's splitting and reproducing. Excellent. Zion, looks like it's multiplying. Gabe, multiplying. Yes, it is. What about D? If I wanted to give it a D word, a word that starts with a D, what word would you say? D. It's a mathematical word. Duplicating. Excellent, Jasmine. There's another word that starts with a D that's a mathematical word. Dividing. Excellent, Jasmine. Yeah, it looks like it's dividing. It looks like it's breaking or splitting. Cells cannot grow in size. They have to grow in number. They have to grow in number. They can't grow big. They can't do it. So what is the cell cycle? These are points that you should be writing down. And you're going to need to use these points later in the exit ticket. There is a grade, and the grade is going to be at the very end of class, not later tonight. It's going to be in class today. So you do need to write this down. So the sequence, what we call growth and division. There's that division, that term that I was looking for. Looks like the cell is dividing. And we can break the cell cycle down pretty easily, but let's think about why we would have a cell cycle. So it's really about growth, right? So if you're a baby or an adult, and here we can see fly embryos have cell cycles every 8 minutes. So if I start out with a cell, and for a fly embryo, you go from 1 to 2 in 8 minutes. In 16 minutes, I go from 2 to 4, and then I'll go from 4 to 8, 8 to 16, 16 to 32, 32 to 64, 64 to 128. It just grows really, really rapidly. And think about it. What's going to grow more quickly? An embryo, which is going to be grow into a fetus or a baby. Would you think an embryo would grow faster or an adult? Which one would have the cell cycle faster? So baby or adult? Drop it in the chat. Babies or adults? Which ones are going to have a faster cell cycle? Thank you, Vu. Thank you. And you're giving it to everybody. Excellent. Jasmine, thank you. Food, thank you. Gabe, everybody. Yes. Thank you. Baby's job is, God, I love it when you guys are doing it. Killing it, Baptiste. Killing it. Yeah, so you guys, baby's job is to get big before something kills it and eats it, right? So predators look for little babies. If you've got a wolf looking to kill something, it's not going to go for an adult. Adults are risky. Kill the baby, and potentially it will be more easily taken down. So baby's job is to grow. When you look at this, you can see liver cells divide. It takes a long time. This is one reason it's really good to avoid compounds like such as drugs and alcohol, because your liver is what has to deal with any kind of compounds that are toxic to your body. And you can see your liver takes a very long time to do what we call regenerate, to grow, for the cells to divide. It takes up to a year. Other organs are much faster. Average for our body, if you take all of these organs, is 24 hours, but your stomach is one of the most rapidly dividing organs. And part of the reason is all those acids. Your stomach's constantly being broken down because you're digesting food, which is made of biomolecules, right? Food's made of biomolecule cells. Well, your cells are too. So you digest your own stomach slowly every day. So you, on average, get 11 stomachs a year. You have to grow new ones constantly. Pretty cool. Cell cycle is going to do that to replace damaged tissues. Cell cycle is broken into two parts. This is important. You will be tested over this today. So you've got this interphase. So interphase is going to be comprised of three parts: growth one, which is we call G1, S, and G2. And these are really easy to remember because G starts, growth starts with G. And these are all centered on growth. And this is going to be the growth phase where your cells are just doing their job, doing what they normally would do, which is basically protein synthesis. And I'll show you what I mean. But then the other part is going to be meionic phase. So when you look at this diagram, interpreting diagrams, you've got this one circle, or one something that almost makes a complete circle, and that's interphase, which is broken into three parts. And then there's the meionic phase, and meionic phase is broken into four parts. And I can't show you all four parts in here. I'll have to show you those separately. And then there's the active division itself. Cyto, which is going to be cell, and kinesis, which is movement. So if you're a kinesiology major, you're a movement major, you're a body major, essentially, a sports major. Interphase, again, means between phases. So the cell is, doing its normal life function. So you got growth. That's going to be protein synthesis. And synthesis itself means to make. And you're in S phase is where you're going to make copies of your DNA. You're getting ready to divide, but you're not dividing yet. You're still carrying out normal functions. And then this would be kind of what it would maybe think about it this way, it'll look like. So you have a chromosome. This would be a chromosome, very condensed DNA. And I make a copy of it. I want to make sure all my cells have an exact copy of my DNA because I want them to have the exact same function. I don't want to grow some other cell out of a skin cell. I want my skin cells to be human. I don't want them to be mice, right? So when I replicate my DNA, it needs to be an exact copy of that DNA. And we'll call these chromosomes that are the same as each other as sister chromatids. They're held together by this thing called a centromere. A centromere is kind of like a button. Think of it like a button holds things together, or like a hubcap holds the wheel, kind of on the center of the wheel and holds the wheel to the car. So think of it that way. It's just holding these two together to keep them organized. You want to keep these things very organized so when we separate it out, it's not broken apart. Second, growth phase, G2, is where your organelles have to double. So if you're going to get a new cell, well, you need new mitochondria, you need new endoplasmic reticulum, new Golgi apparatus. You need all those organelles to be replicated so that they can function properly. The new cell can function on its own. It would be like if I replicated myself, I want to make sure that when I replicated myself, I had four eyes before I divided. So each of me had 2 eyes apiece. Or I replicated 2 mouths or another mouth. And when I separated, each of me had another mouth. Then that would be G1 for protein, S for DNA, and G2 for division. So I didn't ask anybody to tell me to hush. So let's see. Nicholas, can you set a timer for 20 minutes and tell me to hush? Say, hush, Mr. Pope. Can you do that? Actually, set it for less time than that. Set it for 15 minutes. You got me, Nicholas? Zion, you got me? 15 minutes is all the time I have? Yes, no, maybe so. You got to tell me. Okay, I'm going to go to Baptiste. Baptiste, can you set a timer for 15 minutes and then you can come on and tell me to hush? Yes or no? Baptiste. Baptiste, can you tell me to hush? Say 15 minutes. All right, thank you. Set the timer. Awesome. All right, excellent. So there is an acronym that Dr. Smith has just put out for you. It's really called, it's PMAT. And before PMAT, you've got this phase. Again, we just looked at it, interphase. This is the between phase. There's not really anything going on for division. The cells carry out its normal function. And some evidence that it's going to be like that is just that you can see a nucleus. You can see the DNA is all unwound. It's doing its normal job. These are called centrioles. They're not on anywhere, separated. They're right next to each other. So this is a normal cell under normal function. It's not growing. It's not doing anything. It's just, it's not doing anything centered on growth. It's just, centered on its job. Mitosis is really going to be, again, it's about that growth, but it's also about reproduction. Some organisms are asexual. They don't have sex. They can't have sex. They're a cell. Cells don't have genitalia. If you can ever find a cell that has, some sort of female or male genitalia, penises or vaginas, please let me know. That would be tripped out. You will have discovered something completely novel and new. Single-celled organisms, they can't have sex. So if they can't, they have to reproduce somehow. So they just do it by simply dividing, but they need to do it in an organized way. So A means not, and then sexual. Asexual, not sexual. So they grow without having sex. They reproduce without having sex. Mitosis is going to be one way eukaryotic single-celled organisms actually reproduce. And that's going to be nuclear division plus what we call cytokinesis. Cyto, cell, kinesis, movement. So again, if you're a kinesiology major, you study sports. In sports, we move. That's why I don't think that esports is really a sport. You're sitting on your butt playing a video game. That's not sports. That's not kinesis. You're not moving. You're just sitting there. Your thumbs may move, I guess, but that's about it. So the result of this is pretty straightforward. It's going to be two genetically identical daughter cells. The cells that come out of this are called daughter cells. There's a parent cell that produces two cells called daughter cells. These daughter cells go through mitosis, and mitosis, just like Dr. Smith put in there, it's a mnemonic, easy to remember, PMAT. And this is in order: prophase, metaphase, anaphase, telophase. Really, really straightforward. Dr. Smith just dropped some more information for you there. It's excellent. Thank you, Dr. Smith. Now, let's do a little algebra. So if I'm in mitosis and I'm a human, and you do need to know this, you have what we call a ploidy number. So if n equals 23, and this will be something important for the next lesson, if n equals 23, what does 2n equal? What does 2n equal? It's just multiplying. 2 times 23. Drop it in the chat. Drop it in the chat. What does 2n equal? Drop it in the chat. I've got 1. We should have a total of 21 answers. There's 2. 3. Excellent. No, not squared. Multiplying. No VU. No VU. Try again. Thank you, VU. Mario, good job. Yeah, it was a typo. I figured it was. Excellent. Thank you, Mario. Keep it up. Keep it up. Keep it up. Looking for others. Looking for others. Right now, I've got 1, 2, 3, 4, 5, 6.
I have Zanette on my side.
8, 9. I've only have 12 like last time. There we go. There's Sabina. Good. Jasmine. Excellent. Delamore. Baptiste. Good job. You're the boss. You're the boss. Good job, Zion. Good job, Zion. Y'all the boss. Good job. Ugh, Jasmine. Good job, Tavion Graves, man. All right, good job. You guys are doing well. Yeah, it's easy, right? It's just 2 and 46. So I need to replicate this DNA. I need to replicate all 46 of my chromosomes. Sperm and egg only have 23 chromosomes because they have to combine it. We'll get to that later. Prophase, let's look at the first phase. And this is where you really need to be writing down information or you're going to be stuck. So prophase is the first phase. There's some characteristics of this cell that you have to know for the assessment. So first thing I'm going to notice is there's going to be these little structures, these little fibers, these little lines. They're called spindles. These these structures here are called centrioles, and your spindle fibers are connected to them. They separate at the poles. And you want you to think of it like you guys are in geography. You've got the North Pole and the South Pole. At the top, you've got the North Pole. At the South Pole, you're at the bottom. And they're going to move to opposite sides because these cells are going to separate. They're going to pull apart. They're going to pull apart and make 2 new cells. You'll also notice that the nucleus is breaking down. It becomes visible. You can see these sister chromatids. You can see these chromosomes forming. They're condensing. They're organizing. I've got to organize them because I've got to divide them. I don't want to divide unevenly. I need to divide these out evenly. So I take all my chromosomes, or my DNA, I replicate it, and then I organize it. I'm going to separate it. This will be characteristics of the first phase. I'm pausing for just a second so you guys can get this down. You will not be able to do the assessment without this information. When you do the exit ticket today, you can use this information for the exit ticket. It can't stop you from using your notes. Obviously, you're at home in another room. So it's kind of like a little cheat sheet, but you've got to be writing this down. I can't pause and just be like, oh, okay, I'll let you write this down for 10 minutes either. So you need to be taking this down as we're going over it. So prophase is before. Pro really means before, the before phase. So you start with interphase, and then we get to this before they divide, before they separate, before the chromosomes start to pull apart. So these are the characteristics of how you notice that. If you didn't get it down, you can come back and look at the PowerPoint. I've got it attached, but you need to get the next set down because we got to move. So next is easy. It's the really easy way to remember it. It's M for middle. So when you're in geography, you got the North Pole and South Pole. What's that line across the middle of the Earth? What do they call that line across the middle of the Earth? And now I'm going to start picking on people individually. So I'll go to Zach first. What's that line across the middle of the Earth, right down the center? So usually it's,
The equator.
Yeah, I just put it out there. Shoot. Yes, the equator. Usually tropical forests are going to be along the equator. You'll notice your chromosomes are going to line up at the equator, at the middle. Hence, what we call it metaphase, middle phase, M and M. The equator, that's where that's a piece of evidence that you would have. And on the STAR exam, you're going to have to be able to know what these orders are. On today's assessment, you're going to have to know what these orders are. This is your information that will allow that to occur. It will allow you to be able to make that judgment, make that decision. The next phase is the anaphase. And ana is easy. A, away. Anaphase starts with an A. Away phase starts with A. My sister chromatids, my chromosomes are moving apart. They're going away from each other. Goodbye. I miss you. I will see you on the other side. No, you won't. You won't ever see each other again. They don't even have eyes to see one another. But my evidence is it's the centromeres that are holding them together. They break. Spindle fibers are holding those centromeres and they pull them away. The away phase. My chromosomes are moving away from each other. The sister chromatids are separating. They're moving to the opposite poles. So like a north pole and like a south pole. Obviously it's not north and south, but the opposite sides. They're being moved away to opposite sides so my cell can then form two new cells. A parent cell can form two daughter cells. That would be the away phase. And then finally, before we divide, we get to something called telophase. Telophase is really easy to recognize. You got two new, what are looking like maybe going to become nuclei. The chromosomes are starting to unwind. They're starting to relax. They're starting to go back to the normal state. Spindle fibers are formed. You've got something called cleavage in the middle of it. To cleave something is to cut something. My mother used to tell my sister, not to show too much cleavage between her breasts. It kind of looks like there's a cut between these two, kind of like there's a cut in a shirt, and then you don't want to show too much cleavage, and there's this cleavage furrow. And sometimes you'll see it referred to on exams as a cleavage furrow. So it's cleaving, it's pinching inward. So this is little information that you can use to determine this. You'll see nuclear membranes forming. And before we didn't see that. I'll even back up and show you. There's no nuclear membrane. There's no membrane around a nucleus. There's no nucleus visible here at all. But there is here. You can see there's a nucleus. And the chromosomes are going to start to unwind. They're going to start to separate. They're going to start to unwind so they can do their job. They can't do their job when they're chromosomes. DNA can't get ready to synthesize proteins. It can't replicate. It cannot synthesize RNA and then RNA make protein. So you can't do that yet, not when it's in chromosomal form. And the only difference between animals and plants is simple. It's surprise, surprise. It's animals that are going to pinch inward with that cleavage furrow. And plants they can't do that. They've got cell walls. So they're going to have a membrane in here, but they'll also have to form a cell wall. And it's really easy to notice because it's very symmetrical. And they'll have this no cleavage furrow. They'll just have a cell wall that forms between them. It's very, very straightforward and noticeable. But when you can see here, there's clearly a nucleus in cytokinesis. Cyto is cell, kinesis is movement. So they've already broken into 2 new cells. Now let's do a little check for understanding. Cytokinesis, you can see they're moving apart. These are now moved apart. Looks like the membranes are barely sticking together a little bit. They're starting to, these lipids are pulling apart. But let's do this. First person, and you do have to come on your mic. I know Batiste is not scared. He's already proven that. So Batiste, when I'm done with you, you call on the next person that's going to go. So I'm going to ask you guys about these cells, and you tell me what phase of the cell cycle they're in. So Batiste, let's do this one. You'll start here with you. What phase of the cell cycle is this cell in, Jeremiah? What phase of the cell cycle is this cell in? And you don't have to be right. You just have to take a chance. You just have to take a chance. And come on your mic if you can. And if you put it in the chat, you got to chat it openly to everybody. Take a chance.
This movie is not even sad. This is
What do you think? The DNA is pulling apart. It's moving away from each other.
I don't know how to say it, but it's like a,
Okay.
it's a CYTOKIN.
That would be cytokinesis.
Yeah.
Now, but in cytokinesis, you'd have 2 new cells. This is where the DNA is pulling apart or away from each other. It's the away phase. And your options are
Telophase?
Almost. Let me go back and show you. Almost. You almost got it. The away phase is anaphase. Anaphase. On telophase, you would see 2 clear nuclei and it would be pinching inward. You'd have that pinching inward. Let's try another one. Who's going to go next, baby? You choose.
Jasmine.
Jasmine. All right, Jasmine. What phase of the cell cycle do you think this one's in? This cell right here, Jasmine.
I think it's metaphase.
Metaphase? I don't think so. They would be chromosomes would be lined up along the equator. Doesn't look like my nucleus is still formed here. I still have a clear nucleus. Metaphase doesn't have a nucleus. There's still a nucleus here. So what else could it be? And
Prophase?
Yeah, prophase. I think it's either prophase. It could potentially be interphase between phases, maybe between phases. It looks like there might be some chromosomes condensing in there. Good job. Who goes next? You decide. Who goes next? Hurry, hurry, hurry. I want to give you time for the guided practice. I got some people requesting. Zion's requesting. Are you okay with them doing it? Oh, no, you had somebody in mind too. Zach, all right, Zach. No, Zach. Yeah, Zach's already gone once. Can you give somebody else a chance, Zach? I've already picked on Zach.
Okay. Gabe.
All right, Gabe. So what phase of the cell cycle do you think this cell is in right here?
I'm gonna say cytokinesis.
Yeah, it's kind of hard to say. It's either cytokinesis or it's anaphase or telophase. Excuse me, I think they're still kind of connected right there. You can see like there's not a membrane that's formed between them. So maybe this is, I would say, it's transitioning from telophase to cytokinesis. Good job. Who's going to go next?
Mario.
Mario, which part of the cell cycle is this cell in? Is this cell? What part of the cell cycle is this one? Looks like I got
Metaphase?
centriole. Yes, metaphase. Look like I got centrioles on either side, on the opposite poles. I can see my spindle fibers grabbing the centromeres that are grabbing my sister chromatids. My sister chromatids and chromosomes are lined up along the equator. That's clearly metaphase. Who goes next?
Brontavion. Oh wait, did you already go?
Tavion Grays. Bertavion Grays. What do we have here? What about this one, sweetheart? What about this one here? This cell here, Bertavion Grays. Bertavion's a brave kid. Hence his name, Bertavion. What about this cell here? What about this cell? You're supposed to be telling me to hush. I gotta hush at some point. Okay, well, you just gotta take a chance. You just gotta take a chance. You've got interphase, prophase, metaphase, anaphase, telophase. You've gotta take a chance. Interphase, prophase, metaphase, anaphase, or telophase. You've gotta take a chance. You don't have to know, you just have to take a chance. You only have 5 options. Well, you got 6 if you want to consider cytokinesis. Yes, the person that just sent me a chat, yes. You just got to take a chance. We're stuck on you, Bertavion. Stuck on you. What was it? I couldn't hear you.
Metaphase.
It kind of looks like it's getting out of metaphase and going to anaphase. This was a tricky one. I wanted to give you one that was tricky because I know that you've got guts. It looks like it's coming out of metaphase and going to anaphase. They're still kind of connected right there in the middle, right? So I was wondering if you were going to say metaphase or anaphase. Good job taking a risk on that one. If I had to err on one side, I would say probably anaphase, but it's still connected. So
The timer stopped.
Oh, thank you. Well, tell me to hush. You gonna say hush or not?
Hush.
Thanks. All right, then rapid fire. We'll just do this one. So I'm gonna go through 3 more examples and then I'll feel confident that you guys are ready for it. Let's see. Hudson, what is this one? Hudson, what is this one? Hudson, this one's yours. You got interphase, prophase, metaphase, anaphase, telophase.
Anaphase.
That's anaphase. Good job, Hudson. Woo! Okay, so what about this one? Let's see. Muse, Asiya. What is this one? Interphase, prophase, metaphase, anaphase, telophase. Hurry, Muse. We're going to get 2 more before I feel like we're confident enough for people to move on. Asiya or Sia, that's what I'm waiting on. Waiting on you, waiting on you. We can't go to the guy to practice unless you give us something. We're stuck like Chuck. You got to take a shot. We're stuck. I'm assuming you're not here. If you do not answer in the next 15 seconds, I'm going to bounce you from class. I'm sorry, I'm just assuming that you're not here unless you take a chance. You got 30 seconds to take a chance. Oh, thank you, sweetheart. You took a chance. Metaphase, yeah, metaphase. So it is going to be metaphase. It's not quite anaphase. Anaphase, these sister chromatids would be separated. They're lined across the middle. They're lined, these chromosomes or sister chromatids are lined across the equator or the middle. So this would be metaphase. Last one before we do this is going to be Kenneth. So Kenneth, what phase would you call this one? We've got centrioles formed, moving to poles, spindle fibers, and then the nuclear membrane is breaking down with chromosomes visible. What would this one be, Kenneth?
Prophase.
Prophase, good job. All right, I'm gonna stop sharing. I'm gonna show you where your guide and practice is. You guys are gonna have 15 minutes. Oh, I love your superhero, Bautista. Is that your superhero or is that a ball player? Because it's superhero day here at school.
It's both.
It's both. Who's the superhero, dude?
It's Spider-Man.
It's Spider-Man. All right, good job. All right. Okay, so here we are. So when you're going to go to the website, go ahead and go to the website, and you're going to open up today's lesson, which is going to be October 30th, and it's ordering and describing the cell cycle. Open that document. I'm going to choose one of your documents in just a moment, and I'm going to do the first one. If you want me to do the first one for you, chat to me that you have it open. Send it when you get it open. First one that sends me a chat that they have it open, I will do the first one for them. And it's ordering and describing the cell cycle. So first one that gets it open, send a chat. I've got it open, or say I've got it open and your name. Ooh, crud, there's a bunch of people all of a sudden. Okay, Vu, you're first. All right. Vu's like, I want to be the first, man. Okay, let's see. Vu, where's Vu? Down here, question mark. She doesn't even know where she is. There's a question mark here. Where am I? All right, so let's open this document for you, and I'll do the very first one. It's very straightforward. This is an easy one. Several people got this finished last class. You're gonna have 15 minutes to do this. Now, here's the deal. When we go to a breakout rooms, you have the ability to share screen. You need to share your screen. One person be sharing their screen, and other people help. Other people helping that person order it. One person can go back and look at the notes, and then you can help somebody else to order this and get this straight so that you can go back and do yours. There's different ways to learn things. You can listen, which is one way, but that's what most of you guys are, only way most of you guys are doing this. You're just watching us, and that's not enough. You can write, that's another way. You can manipulate work here, that's another way. You can talk, and that's another way. So if you talk about it, write about it, hear it, speak it, you learn it. To just simply watch it is like watching TV, and that's not going to get you where you need to go. So I'll do the first one. You can see I've got several of these in order. So I've got interphase. That's going to be the first one. And then prophase, metaphase, anaphase, telophase, and cytokinesis. These images are going to match one side over here. So when you're done, this side is going to be blank. These images, it says up here, images to be used in left column. This is the left column. This is the right column. This column will be blank when you're done. So I'm going to take this image. I know because I've built this. This is going to be prophase. So I'm going to click on this and cut it. It's gone. I'm going to put it here. And you'll do the same thing in a moment for yours. And I'm going to put two pieces of evidence. Evidence that I gave you. So evidence that I see is there's a nucleus, or a nucleus is visible. It's visible. And these little structures, these centrioles are not at poles. They're together on one side. This is what a normal cell should look like. The organelles aren't in there other than the nucleus, so it just makes it easier. Then what you've got to do is do the other one. So this is going to be prophase. This is going to be metaphase, anaphase, telophase, cytokinesis. And then there's two questions. So I'm going to drop you guys in breakout rooms. And I'm going to get, when I come to your breakout room, I'm going to need to see one of you guys sharing your screen and everybody having their document open. So you'll be working on your own document and as well as going back and looking at the PowerPoint that I just went over. I want you to go back into it. It's not enough just to casually do this and answer a couple of questions at the end. You need to think. All right, here you guys go. You're in breakout rooms. We're going to put the most beautiful children in the first breakout room. Oh, no, there's nobody to do that. I can't put anybody in there. That's terrible, right? Okay. So. Boom. Some of the breakout rooms are open. So. When you get a breakout room, join it. When you get a breakout room, join it. Okay, let's see how you guys are doing over here. Who's going to share their screen? Who's my screen share? You guys got to talk, got to talk.
I could, I guess.
Okay, do it. Let's do it. Let's start working. Let's make haste.
All right. Here we go. So,
Awesome. Now you got to start working on this, and somebody else is going to be a person that's going to pull up the PowerPoint and talk your way through this. You've got to talk. You've got to talk. Everybody should have their document open, but one person should have the PowerPoint. At least one person. Talk your way through it. I'm going to bounce out. If somebody just got kicked out, dropped from the call, I got to put them back in the room.
Join a room, okay?
Yeah.
Mr. Pope,
Yes, ma'am.
can you activate the share inside? You did it for the last class. It's the option for them to be able to share inside of the
Oh.
breakout room.
Oh, they're able to do that. Yeah. I went the last
Okay.
class. The last room I went in, they were all working together.
So when I open it in my screen this time, compared to last time, it doesn't show that option being activated by host.
I just came from group 5. They were doing it.
Okay, it was showing something
And
different.
it shows me on too. It says allow participants to, and it says share screen chat, rename themselves, and unmute themselves.
Yeah, under the advanced sharing, it says who can share. It says, okay, so I do see it on here. Let me go back in and see what's
Yeah, it's working
going
in
on.
group 5.
Okay, let me go back.
All right, let's see. Who are we working on? Who's sharing the screen?
Oh, I am.
Oh, okay. Okay, so let me see what you got. So what is this first phase here above? What
That
is
one's the pro phase.
Oh, that's
And
interface.
then
Nope, that's
This one?
interface. Yep, that's interface. You can put it underneath the interface if you want. So you kind of got them a little out of order there because you went from interface to metaphase. That can't be right. No, that's okay.
Okay.
Interface is
Oh,
first.
spindle forms.
Yeah, interface is first. Interface is first. It was in the correct spot. It's just that
Oh.
you just put metaphase after interface, and that's not true. There you go. Get that sucker out of there.
And then this one, move it.
Yeah,
down.
get that on out of there. Prophase is going to go there. Oh, yeah, you got it.
Oh.
Right. Oh, look at you. You know what you're doing. All right. Who's helping you do this? Who's got the PowerPoint open?
I
and
have-
talking through it?
Oh, I don't have the PowerPoint up.
You just have your notes?
Yeah, I have
Okay.
I wrote all the.
Okay, so good. You should be asking them questions though. Make sure that they're giving you information. Don't let Y sit there. Don't let Zach sit there. Don't let Zion sit there. But make them engage with you. I got to check on other rooms. I'm bouncing.
Okay.
Yeah, it should be able to. So
Yep. I just
There's
came from
the
3
interface.
rooms and they're all sharing screens.
Yeah, they're saying that they don't have access, and I see on my end now that it shows access, so I don't know what's going
They,
on.
they have access unless this is the only room in the world that doesn't. The other groups do. They've got to go down to that share screen button, hit it, and they should be able to share screen. I've just come from literally 3 rooms that are doing this.
I pushed the button more than once and it keeps saying host disabled screen sharing.
Really? It's so weird. All right, hold on a second. Let me see. I can't do anything from while I'm in this room. I have to leave the room to change settings. Let's see. Okay, who is talking?
I mean,
Is that
I
Bert-
can share my, that was that Bertavian? Because Bennett volunteered to share, and then he said he couldn't, and Bertavian, I think it was Bertavian that was saying that, but they were all saying the same thing.
Okay, you know why I bet it is? I bet it's because you're in here. $100.
Really?
That's gotta be the independent variable.
Okay, well. I'm
You know what I
gonna go-
mean? Big, I
Well,
bet
yeah.
that's it. I bet it defaults to us when we come in here with them, but if we leave, they
Yeah.
try.
Okay, because they were able to do it last class, and I was in there with them the whole time and walked them through it, but I'll leave and see.
Yeah,
I don't
let's bounce out and we'll bounce back in. Y'all
Okay,
try
so
to
while
do
we're
it with
leaving-
when we leave,
Yeah.
and then we'll bounce back in a second. Uh-oh, we got 2 kids outside of the rooms.
Yeah. What's going on, you guys?
Hello, Mackenzie and DeCorian. You guys got out of your room? Hello? Oh, they just joined the room.
One of them there, Mackenzie's still here. I
Yeah,
didn't get
Mackenzie's still
assigned.
here.
She said, okay, saying that she didn't get assigned to a room.
What? No, she's assigned. You're assigned to, all you gotta do is go to group 5. You're assigned to group 5. Jump in. Yeah, I'm looking at it. You are assigned to group 5. All you gotta do is join the room.
So like a little screen would have popped up, so,
I can assign you to another room and see if it, no, there she is.
okay.
No,
All right, so-
she's not. She's gone.
Yeah.
She just
I'll
disappeared.
go back and check on, what was that group?
Yeah,
one?
see if they're able to do it now. Maybe they've had some time to get it now. I'm gonna go check on, there's only one room I haven't been in yet.
Well, I can technically only go to one room, and I don't know why that
Oh,
is,
really?
but remember I told you last time,
Yeah.
it has to be something in the settings, but it's now only let me, I can't maneuver between rooms.
Okay, I'm sorry. All right. What room are you going to? Okay. Over everything. Okay, what do we got here? Hold on a second. There, well, sort of. What happened? Hello? Who's sharing their screen in here? It won't let you in the dock? Huh. I'll go check. Are you logged in as you're on your Uplift account? If not, somebody else share their screen and do it. So it's either going to be Vu or Brasinho. Huh. I don't know why it won't. I'll go check on yours. I'm going to stay in this room, but I'm going to go check on your document. Make sure that you can open it. So let's see. Back. Somebody else in the meantime, jump on theirs.
Wait, Mr. Pope. So I'm
Yeah,
trying
I'm
to
listening to you. Uh-huh.
right now, but you know, in the first one, there's like the purple thing is the nucleus, and then like the 2 pointy thingies is the
Centrioles, yeah.
centrioles. So for like the second and the third one, I haven't organized them yet, but the centrioles would be the light brownish ones, right? It wouldn't be the red or blue ones.
Oh, you don't use
Purple.
colors. I'm colorblind. That's terrible for me. I'm gonna show me the images with your cursor. No, they're directly when you say
this
red,
one,
blue.
like-
On it. Those inside.
Okay,
Those are
so
those are going to be chromosomes or sister
Uh-huh.
chromatids. Those are sister chromatids. Chromosomes. That's
so this
spindle
is the one
fibers.
that we're looking about, right?
Those are spindle fibers. Yes, those are spindle fibers. The
So
centrioles.
what we're like answering is about this one.
Yeah. You got to give me some characteristics. What phase do you think that is?
Which one are the centrioles? Is it this one?
Yep,
Like
yep, that's a
that one
centriole.
right there?
Yep.
Oh.
Those are kind of like anchors. Centrioles are kind of like anchors. They actually anchor to your cell membrane, and then they have those spindle fibers, kind of like ropes, and they pull those chromosomes apart, those sister chromatids apart. They haven't grabbed the centromeres on the chromosomes to pull them apart yet, though. You don't need to know that much as a ninth grader, but you just doing a little extra info. So you've got prophase. That's prophase though. Prophase is going to come after interphase. So go up. Let me see what you have on the second phase. Yeah. So prophase goes there. That's anaphase. That's you're going to have to change
Okay.
that position.
So the order that I had before was right.
I don't know. I don't know what order you had before, but that's the second one right there. That's prophase.
Okay, I know these are the last two because
Yes, they are.
they split apart.
Yeah,
And then the
exactly.
top one's right. So you're just saying I need to switch them. Okay.
That top one's right, and then the next one's going to be prophase, and the one that we were looking at, that one right there. Stop. That's anaphase. So that'll be the fourth one.
Okay.
You go interphase, prophase, metaphase, anaphase. And Beck, I went and checked your permissions. I gave you extra permission. Like I granted permission twice for you. You should be able to open it. Now, somebody needs to be pulling up the PowerPoint and giving you extra information. What's your evidence that this is prophase? What can you tell us that this is, why is that prophase? And I'm going to bounce into another room and we'll come back to this in about we'll leave here in about 4 minutes. Get as much as you can done. Scare me to death. I want us to bend in my step and get the better over everything. All right, let's see. Who's sharing their screen over here? Woo! Mario, excellent. So Mario, let me help you out for a moment. So the first one is prophase. You got that. That second image you have below that, no, excuse me, the first one is interphase. That second one you have there below it, that's metaphase. That's third. You go interphase, prophase, metaphase. There you go, get it out of the way. Put your image for prophase in there. There you go, that's right, that's prophase. Get that sucker in there. Good job. All right, so what comes after prophase? I hope they're watching this because you're doing a great job. Woo! Well, hold on a second. I think you jumped the shark there. There you go. You're just so fast with your fingers. You guys are just like lightning bolts. Okay, cool. Metaphase. Now we got to get, that's telophase and cytokinesis. We got to get them out of there. Got that cytokinesis. That's last. Excellent. That's telophase. There you go. Now look at you. You're like a lightning bolt. Whew, man. You got to be Usain Bolt as fast as you move your fingers. All right, now you got to start filling in characteristics. That's what you got to do. Get these. What's your evidence that these are the phases they are? So you other kids, Jeremiah, Jasmine, Jordan, and Kenneth, are telling him what to type in there. Because you got the order correct. Now you got to get evidence that that's why the order is the way it is. We only have 2 more minutes. I'm going to bounce you guys out of here. See you in a sec.
So where do you have notes, Batavian?
So Bertavian's able to share his screen?
No, this is me sharing my screen, but he's in his, and so I'm just talking him through it.
Oh, okay.
Because we're still not able to. So you
Oh,
guys
wow.
are giving me one bullet point. So what's another bullet point? So go back to your notes as well as support. So
Yeah,
what
and
else do you see here? So Oh, go ahead.
I was just saying, we've got to come out in 2 minutes. The other groups are almost all done. They were able to share screens. I don't know why this group couldn't, but the other groups are almost all done. So what we've got to have time for exit tickets. So I'm going to bounce everybody out in 2 minutes, 2 minutes.
Okay, thank you. So you have.
What's that, baby? Oh, man. How old are you, kiddo? Oh, no. Just, if there's somebody out, wait for them to come back. Don't ask, don't wait for me. Just go to the restroom. They're 17 years old. It's ridiculous. Who was in here before? Mr. Andrews? Not that formal. It's Friday. Hey. Everybody wants to get down. It's Friday. Hey. Rosino, don't you usually have like soccer balls with a curtain behind you? Like of a sports curtain behind you? Is that you? You're muted, kid. You gotta put your mic on.
Huh?
Don't you usually have a curtain with like soccer balls, footballs, basketballs and stuff behind you?
Yeah.
Yeah, I was like, where is she today? Once everybody gets in here, we'll show you where the exit ticket is. Wow. All right, everybody. You guys did a pretty good job. I looked like whenever I bounced in and out of your rooms, that 3 of the 5 of your groups got that, at least got one document for their common working together, got one done. So I think the lesson to take away from this is teamwork, right? When you have multiple people working together, you can get these things, these items done. Maybe even think about doing that before you take assessments. You can make your own Zoom links and have meetings where you guys work on your homework together and study together so that you can get a lot better at this. Right now, we've got to really do all we can to get ready for the academic year, guys. We still have 3 quarters to go. So I want you to go back to the site. Let's see, let's go back to our class site. And you should see our exit ticket for today. Now the exit ticket, I'm putting it as a scholarly habit. It's only worth 10% of your grade, these exit tickets. They're not formatives. They're not summatives. So these, in terms of your exit tickets, we're not doing retakes. Do not go in there and just click, click, click. You can retake formatives, you can retake summatives, but these exit tickets, I want you to take them seriously, but they're not going to be something that's going to radically change your grade. But you pay attention in class, and if you are scholarly, you take notes, you'll be able to use your notes on these. And there's nothing on here that you can't get with your notes. It is time, it is 5 minutes. So I need you to open it and take it now before we leave. And then I won't see you till next week. I'm gonna miss you guys. These Jasmine will smile at me sometimes. I won't see you till Tuesday. Now she's smiling now. Yay. Yep, so we just read the questions carefully. And I'll go and I'll open, there's a question about one of the questions. I'm gonna go and see if there's something weird about a question. Yeah, you'll just have to think about that. These are the questions. Of course, you don't see the images, but maybe. Yes, when you're done, you can leave, but take your time on it. Don't just try to rush through it. Oh, it's not a big grade. It's something. And it's really the difference between A or B or B or C, that kind of thing. Have a nice weekend too. Thank you. The document that we posted here is due tonight at midnight.
Okay, what's it called? I'm done, by the way.
Yeah, I saw you were done. Yeah, the one
Okay.
that's here, there always shows due dates on it. So anything we ever give you shows the due date. So tonight about midnight, and I wasn't able to catch up on cellular respiration yesterday. I will dedicate my evening to get all those read. So let's get all these
Okay.
done tonight. Thank you.
What's it called? May I leave?
I'm sorry, baby.
May I leave?
See.
Okay, thank you.
Adios.
Have a good weekend.
You too, baby.
Bye.
Bye, you too.
Sweet, kind child. That's super sweet. Yeah, you too. Thank you. You have a nice weekend too, Jasmine. You have a nice weekend as well, Zion. Adios, Batavion. Thank you. You have a nice weekend. Spider-Man, Spider-Man, does everything the T's can. Flies around on the walls, does everything except for fall. Spider-Man, Spider-Man,
I
Jeremiah.
think you're missing a few words.
I don't know the song, man. I forgot all this stuff. I haven't watched cartoons in 30 years. I just remember Spider-Man, Spider-Man. That's all. By the way, this morning, there was a beautiful owl on campus. It was gorgeous. I got here really early and it was sitting right by the sidewalk. It was beautiful. Like, it was beautiful.
Take a
this morning.
picture?
No, I didn't take a picture. I just looked at it.
Oh, okay.
It was a barred owl and it was just sitting there looking at me. I was like, hey, what's up, owl? You know, it's cold. It was like, I've got to eat, man. I'm losing heat. Got to find a rat. There used to be a couple of owls on campus. I've collected a lot of their pellets or their poop. I've got a bunch of their pellets in the class. Whenever I teach ecology to the students, we take those pellets apart and look and see everything that all the skeletons that are inside of them. All the, like, not I say skeletons, but bones. And they try to put some skeletons together from whatever they've eaten on campus.
I know that is scientific, but at the same time, I'm like, yes.
I mean, it's just, all it is hair and bones. You know, it's all.
No,
they find.
I completely get it, but the visual is just not.
I mean, I call it, and they're owl pellets, right? And I don't even think the kids get it, but then when they look at it, it's just hair packed together
Yeah.
with bones sticking out of it.
It's one thing to like read that, but it's another thing to actually like, oh, I just actually looked at that.
Now, I think they're more disgusted by the reading of it, but I think when they look at it, they're like, oh, it's just a little hairball. I'm like, yeah.
Mhm.
In fact, I'm going to put the Do Now up. I'm going to walk upstairs and see if somebody in the hall will watch the class. I'll go get one and show you.
Oh no,
I need-
Mr. Pope, I'm so
You
good.
don't want to see it? You don't
I
want to see
do
it?
not. I absolutely do not.
Really?
I'm so
A hairball.
serious. I'm so good. I'm so good. There are certain things that I'm good on, and that
Oh.
would be one.
Well, whenever I teach ecology, the kids love doing that. It's really, and they really get it. So in terms of the food chain, so
Mhm.
sometimes we'll find, and you'll find insects in there as well. They'll eat. Sometimes you'll find that the owls will get these big cicadas, and you'll see big pieces of cicadas in there. So you got that in the food chain, and sometimes I wonder whether they found seeds from things that the mice have eaten and that wasn't digested. So they'll see those seeds
Like
from
bits and
plants.
pieces. Yeah.
Yeah, and they'll see like all, you know, since those owls are at the top of the food chain, they'll find all sorts of things in there.
Mhm.
It's really cool.
Yeah.
They're like, oh, wow. And I was like, yeah, well, they're tertiary. They're at the top.
Yeah.
Yeah.
Like
Oh,
I mean,
yeah.
I've looked at the stomach contents. We had to basically dissect cadavers and things like that, but there's still like some things that I still have to process mentally before I do it.
Nobody showed up for class yet.
We're technically still, wait, I always do that
No,
backwards.
12:10 is, like, their class
Yeah.
has been over for 8 minutes.
No. Technically, the next class, oh, yeah, that's right. Because the next class starts at 12:10, right?
12:10, yeah. The last class has been over for
Okay.
8 minutes.
Got it. So are we going to stop this one because we still have two students for this class?
They don't know why they're still in here. I
Hey,
think they're
guys.
just, I don't know.
Wise, DeCorian,
I just think they,
I'm in here. I'm in here. I'm in here.
are you finishing?
yeah,
up
but you need to go to
something, or
your
are you
next
just-
class, yo.
All right, I'm in the last question. I'm gonna go. Do
Oh,
it.
it
It's gonna
doesn't time out?
on you.
doesn't
It does
and
time out.
close?
All
it
right, I'm
does.
gonna go do it. I'm gonna finish it.
Oh, okay. Because
I don't know.
it's been more than 5 minutes unless you just started.
Yeah. Because it'll time out on you. You can't just sit there on your hands.
Oh, I didn't have no time, but I'm finished.
I assure you it had time.
You're doing the right thing, right? You're working on the exit ticket, correct?
Correct. I'm working on the cell cycle ticket.
Okay.
I don't know how. All right, I'm going to bounce you out of here so that I can start the next class.
Do you want to completely close and reset, or for
No,
recording?
no,
Okay.
I'll just stop recording.
Okay.
Thank you for saying that. 'cause-
I'm done. Bye. Thank